Hello students, welcome back to my YouTube channel. I'm Dr. Arpita De. The topic for today's discussion is Kelvin cycle from light independent reaction. In light independent reaction, our focus will be to study Kelvin cycle where we see the formation of glucose. In light reaction, we get NADPH and ATP. This ATP and NADPH thus produced in light reaction is used in light independent reaction to generate carbohydrate. In light independent reaction, our topic today is C3 cycle. Now why do we call it a C3 cycle? This is because the first stable product of Kelvin cycle is a 3-carbon compound. That is why this cycle is also known as C3 cycle. The C3 cycle has got 3 stages or 3 phases: carboxylation, reduction and regeneration. In carboxylation, the atmospheric CO2 is fixed with the help of Rubisco enzyme. Then the product thus formed is reduced with the help of NADPH, which gets reduced here, and then coming to regeneration. Finally, the CO2 acceptor molecule is again regenerated and sent back to the cycle. Now first we'll come to the carboxylation phase. In carboxylation phase, RUBP is a 5-carbon compound which acts as the acceptor of CO2 molecule. Here we see the enzyme is Rubisco and the product formed is 3-phosphoglycerate, which is the first stable product of Kelvin cycle. This is a 3-carbon compound which then gets converted to 1,3-bisphosphoglycerate. Here, Rubisco is a very famous enzyme known to play a dual role. This can mediate reactions with carbon dioxide as well as oxygen. Here, we get to see its CO2 related role. Here, what happens? 3 molecules of CO2 combines with a 5-carbon acceptor molecule ribulose 1,5-bisphosphate. This step makes a 6-carbon compound that splits into 2 molecules of 3C compound or 3-PGA. Therefore, 6 molecules of 3-PGA are formed, which gives a net count of 18 carbons. This reaction is catalyzed by RUBP oxygenase carboxylase, or that is Rubisco. Now, this 5-carbon compound, after combining with CO2, gives 3-PGA or 3-phosphoglycerate, which in turn forms 1,3-bisphosphoglycerate. This reaction is mediated by the enzyme phosphoglycerate kinase, and here we get to see 6 ADP molecules are used. Now coming to the reduction phase. During the reduction phase, this 1,3-bisphosphoglycerate molecule is reduced to glyceraldehyde 3-phosphate. This is done by glyceraldehyde dehydrogenase enzyme. The product formed is also a 3-carbon compound. In the second stage, ATP and NADH are used to convert the 3-PGA molecules into molecules of a 3C sugar, glyceraldehyde 3-phosphate. 6 molecules of G3P are formed. Here in this stage, NADPH donates electrons to or reduces a 3-carbon intermediate to make G3P. Finally, we come to the regeneration phase. Here, the carbon dioxide acceptor or RUBP is once again regenerated. We see glyceraldehyde 3-phosphate is formed in this reduction phase. Now 6 molecules are here. Among the 6 molecules of glyceraldehyde 3-phosphate, 1 molecule goes to the formation of triose sugar. From here it will go, 1 molecule will go, 1 molecule goes for the formation of glucose. The rest 5 molecules are there to regenerate the ribulose 1,5-bisphosphate. Here also in this stage we see 3 ATP molecules are used. So this is the entire picture of our Kelvin cycle. In order for one G3P to exit the cycle, 3 CO2 molecules must enter the cycle, providing 3 new atoms of fixed carbon. When 3 CO2 molecules enter the cycle, 6 G3P molecules are made. One exits the cycle and is used to make glucose, while the other 5 must be recycled to regenerate 3 molecules of REBP acceptor. 3 turns of the Kelvin cycle are needed to make one G3P molecule that can exit the cycle and go towards making glucose. 3 CO2 combined with 3 REBP acceptors making 6 molecules of glyceraldehyde 3-phosphate. 9 ATP are converted to 9 ADP, 6 during fixation step, 3 during regeneration step. 6 NADPH are converted to 6 NADP plus during the fixation step. So this is the entire picture of Kelvin cycle in brief. If you have any questions in mind, any doubt, please feel free to message me in the comment box. Thank you so much for your kind attention.